In this video, we will discuss about another function of calreticulum and calnexin, that is multi-process molecules. It is apart from helping the proteins to acquire their native structure, they also help in maintaining the calcium homeostasis within the cell and also within the endoplasmic reticulum. So, as we are aware that calreticulum plays a important role as a chaperone to helps not only acquiring protein their native structure but also helps in processing or initiation of N-glycan. Apart from this, the it also helps to maintain Ca2 plus uh, homeostasis within the endoplasmic reticulum and also within the cell. So we have discussed about different domains of calreticulum in one of our previous videos, and we have understand that C domain plays a in that video we have get an idea that C domain plays an important role in calcium buffering. So why this happens? Because the C domain of the calreticulum has most of the negatively charged residue, and Ca2 plus is positively charged. As we know that positive attracts negative, so most of the calcium is bound at the C domain of the calreticulum, and calreticulum is a ER lumen domain, ER lumen protein. So it is present within the endoplasmic reticulum, and the capacity is so high that the per mole of protein can bind around 25 mole of Ca2 plus uh, within the endoplasmic reticulum, and it takes care about 50% of the Ca2 plus that is necessary within the ER lumen. And from this uh, we can understand the importance of calreticulum in maintaining Ca2 plus homeostasis within the endoplasmic reticulum. And now we will understand how this calreticulum along with calnexin helps in maintaining Ca2 plus level within the cell. So we have discussed this about the comparison of this to calnexin and calreticulum in our one of our previous videos. However, for this video, we will focus our attention on C domain of the calnexin. The C domain of calnexin also has ER retention sequence, that is RRE, and at the same time it also has negatively charged residues that can bind Ca2 plus level, that can bind Ca2 plus. So what happens is that whenever there is a change in Ca2 plus within the cell or in the cytosol, there is a phosphorylation of serine occurs at 562, that is in the C domain, and that gives a signal or that gives a signal that helps in coupling between Ca2 plus signaling, and at the same time the Ca2 plus containing chaperones within the endoplasmic reticulum, that is calreticulum, and the whatever the fluctuation of Ca2 plus level are there in the cytosol, that will be maintained, or that will be equilibrate by the excess that will be equilibrated by the Ca2 plus that is present in the endoplasmic reticulum, uh, for example with calreticulum. That is why this region is also called calcium buffering domain. So any fluctuation of Ca2 plus in the cytosol will be taken care of by calreticulum with the help of calnexin, as the phosphorylation of serine at 562 position that we have already said that will lead to coupling of Ca2 plus signaling with the Ca2 plus containing chaperone in the endoplasmic reticulum, and this has been uh, tested by in a Drosophila organism by affecting the calnexin in Drosophila, and with this they have identified that there is a fluctuation of Ca2 plus that is not been taken care of as compared to the wild type. And apart from this, Ca2 plus also interact with cell sorting protein. So what that does is that the cell sorting protein distribute the calnexin based on their need to uh, mitochondrial membrane, ER membrane, and the plasma membrane. So that's why from there uh, we can imagine that Ca2 plus can function in different parts of the cell as a signaling or as a say, coupling molecule uh, that helps in coupling Ca2 plus signal with uh, the Ca2 plus containing chaperone in the endoplasmic reticulum. So this is how the calnexin and calreticulum plays a role in Ca2 plus homeostasis, along with maintaining the native structure of the proteins during co-translation and or post-translation, and also helps in initiation of N-glycan post-translation modification. So that's all for this video. Thank you for your time.